cool. I think we're recording now. Cool. So hello, everyone that is here joining us today. Today we have a discussion about the speed of evolution. And before I introduce everyone, I'd like to begin by acknowledging the traditional custodians of this land in which each of us are today, and pay my respects to elders past and present. Today we have two very special guests on the other side of the world. Our first expert on the topic is Martha Munoz, who is an assistant professor at Yale University in the US. And our second expert is Mark Johnson, who is an associate professor at the University of Toronto and the director of the Center for Urban Environments. In our panel today, we have Ben Phillips from University of Melbourne, Lutske Krook from the Australian National University, and we have Celine Frere from the University of the Sunshine Coast. And Belle will be sharing our panel. So thank you all for joining us today. Thanks to our panelists and speakers as well. And I think we're ready. We can begin whenever you're ready, Martha, to start your presentation.
All right. Well, thank you so much, Ileana, for the introduction. Thank you for the invitation to be here, for the panelists, and for all of you that are tuning in Australia. This is a really fun and somewhat surreal experience, and I'm super excited to be here. Ooh. So at the broadest level, my research is motivated by the fact that evolution proceeds unequally. Sometimes evolution is extremely rapid, and in other cases, it seems to stall. So my research program centers around understanding why. Why is it that some traits and lineages get to zip along evolution's Autobahn? And in contrast, why are other traits stuck, in New York City or in Melbourne at rush hour? Now, among those extremely rapid examples of evolution are adaptive radiations. And if you look at these adaptive radiations, many of those exceptional cases of fast evolution are centered on islands or in their aquatic analogs, lakes. And the mechanism that is thought to underlie a lot of these insular radiations is ecological opportunity, this release from competition and predation. And perhaps one of the most classic example of this phenomena is anolis lizards from the Caribbean. On each of the major islands of the Greater Antilles, anoles have independently diversified into numerous structural niches, resulting in these convergent assemblages of morphological specialists termed ecomorphs. Now, central to the adaptive radiation of anoles is this ecological opportunity presented by simplified island biotas. As these lizards were released from their ancestral competitors and predators, they were able to rapidly and prolifically diversify into all of these distinct structural niches. But in the anole adaptive radiation and in other insular radiations in general, the specific role of islands in stimulating evolution is often more implied than directly tested, leaving generally uncertain the role of biogeography in stimulating diversification. Now, in particular, these Caribbean anoles are part of a broader neotropical anole radiation that is comprised of more than 400 species, with new ones being discovered pretty much every other week. Now, anoles are extremely diverse in the Caribbean, with more than 150 species present. And island anoles are, quite frankly, spectacular, but so too are their mainland counterparts in coloration, body size, body shape, and in ecological diversity. So really that's the work of this study, to compare the physiological and morphological evolution of mainland and island anoles, and also examine their rates of speciation. But to get there, this story, there's an origin story here that actually begins on the island of Hispaniola. Now, I've been working on this island for more than a decade, and I was initially drawn to it because of how topographically complex it is. You can go 3,000 meters above sea level and still be on a Caribbean island, but only if you do so on Hispaniola. And in particular, I was fascinated by this radiation of trunk ground anoles, the cybotoids, this lineage containing Anolis cybotes, pictured here, and its close relatives. And I was struck by the elevational diversity that this lineage exhibits. Some species are restricted to low elevation, others creep into mid-elevation, and I was especially struck by Anolis armorii and Anolis shrievi, two species restricted to the highest elevations of the island. Now, all of this topographic complexity of Hispaniola translates into a lot of thermal variation. It can be pretty cold on the island. In fact, up to 20 degrees Celsius cooler at the highest elevations relative to sea level. You can get frost on this Caribbean island, even in the summertime. And through my own expeditions there, I quickly discovered just how thermally challenging these high elevation environments can be. But when I started to look at the field measured body temperatures of these cybotoid anoles, what I discovered is that they're actually really nimble thermal regulators. Across 2.5 kilometers of elevation, body temperature only drops by about 1.5 degrees. Now, to put these data into context, if these same lizards had been behaviorally passive with respect to the thermal environment, body temperature should have dropped by something more like 10 degrees Celsius. And so they're incredibly nimble at transforming really thermally heterogeneous environments, both within and among sites, into ones that become more thermally homogeneous, across the whole island. But such effective thermal regulation doesn't just, shield these lizards from environmental extremes, it also affects their physiological evolution. Through these buffering behaviors like thermal regulation, organisms effectively shield themselves from natural selection. When translated over long evolutionary timescales, such buffering behaviors can actually slow the rate of physiological evolution, a phenomenon known as the Bogart effect, or you might also know it as behavioral inertia. And that's exactly what's going on in these anoles. These diurnal anoles are incredibly nimble at thermal regulation, which shields them from selection on their upper thermal traits. In contrast, cold tolerance evolves up to an order of magnitude faster, reflecting the limited thermal regulatory capacity afforded by thermally homogeneous nighttime environments, which also become progressively cooler with elevation. And I spent a lot of time, teasing apart this radiation, figuring out how they thermoregulate and what was going on. But at the end of these studies, I was sort of left with a bigger question, which is this Bogart effect, this restraining effect of thermoregulation that is so strong in these cybotoid anoles, really, a cybotoid phenomenon? Is it something that characterizes Hispaniolan anoles more generally, or island species? Or is this something that characterizes anoles in general? So the work I'm about to describe was led by Giancarlo Salazar, an undergraduate in my lab. And what John did was gather every datum he could find that it was ever recorded for body temperature, heat tolerance, or the critical thermal maximum, CT max, and cold tolerance, the critical thermal minimum, or CT min, which he supplemented with lots of data that he collected on his own from mainland species. And our first step in this entire process was to simply ask, does the signature of thermal regulation differ between island and mainland anoles? So what you see here on the plot is mean annual temperature on the x-axis, and this is meant to be a relatively crude proxy of the thermal environment that different anole species experience. The field measured body temperature from lizards is on the y-axis, with orange dots corresponding to island species and blue dots corresponding to mainland species. Now, body temperature data are inherently noisy. They're subject to the vagaries of weather and many other factors. But in general, what we found was that body temperatures are higher and generally more stable in island anoles. By contrast, and this is the part that perhaps surprised me even more, is that body temperatures from mainland anoles seem to much more cleanly track the available thermal environment. So this suggests that thermal regulation is stronger on islands than in the mainland. And this is actually exciting because it suggests that this Bogart effect, this restraining effect of thermal regulation on evolution, might correspondingly be stronger on islands than on the mainland. And if so, we had a few hypotheses we could test. First, the rate of evolution should be slower for heat tolerance on islands than on the mainland. These island species might be evolving to a higher phenotypic optimum for CT max. And if it really is thermal regulation that matters, then we really shouldn't detect any difference in cold tolerance between island and mainland species, almost as a sanity check. So what we did was fit a series of evolutionary models to the heat and cold tolerance data. The simplest model was a single rate Brownian motion model in which the phenotypic differences among species are simply proportionate to branch length or time. A slightly more complex Brownian motion model allowed the evolutionary rate to vary between island and mainland lizards. The other three models were all adaptive evolution or Ornstein-Uhlenbeck models that assume the presence of a phenotypic optimum. The simplest model assumed that there was a single global phenotypic optimum for either heat tolerance or cold tolerance for island and mainland species, and more complex models allowed the location of that phenotypic optimum to vary. So in short, there were 5 models, and what we discovered was that the evolutionary dynamics differ between cold and heat tolerance. For cold tolerance, all we need is a single rate Brownian motion model to explain phenotypic differences among species as generally expected. Heat tolerance evolution, in contrast, definitely differs between mainland and island anoles, but does it differ in a way that suggests the Bogert effect is stronger on islands? As a reminder, if this is true, the rate of evolution should be slower and the phenotypic optimum should be higher, and that's exactly what we find. The heat tolerance evolution is 3 to 4 times slower on islands than on the mainland. And the phenotypic optimum for CT max is, about 4 degrees Celsius warmer on islands. Okay, so if you know anything about anoles, the notion that evolution is slower on islands is somewhat weird because anoles, and in particular Caribbean anoles, are, if anything, a paragon of rapid evolution. And the mechanism underlying this rapid evolution, again, is ecological opportunity, this release from predation and competition. So instead, we're actually finding that physiological evolution is slower on the islands. So what gives? Now, mainland and island anoles differ in many ways, but perhaps the most salient for this story relates to movement rates. Now, competition and predation is stronger in Latin America, and anoles correspondingly spend a lot of their time doing nothing much at all, being as visually inconspicuous as possible. In fact, I did my first expedition with anoles to Costa Rica, recorded their behavior, and was convinced anoles just didn't move around. And then I started working on island anoles and discovered, oh no, they spend most of their time running around their habitats looking as conspicuous visually as possible. And this is that hallmark of ecological release. But release from competition and predation doesn't just allow anoles to run around and exploit different structural niches, it also impacts thermoregulation. Because the decision to thermoregulate reflects a trade-off between its inherent benefits, like a performance advantage, counterbalanced against its inherent costs, which often distill down to exposure to predators and competitors. And that's precisely what these island anoles have been released from. So putting this all together, this leads us to the somewhat surprising conclusion that ecological opportunity, a factor almost, so often associated with accelerated evolution, can actually pump the brakes on the whole process by reducing the costs of thermoregulation. So at this point, we started to really wonder, how different are these island and mainland radiations? Do these islands really afford any enhanced context for adaptive radiation to unfold? And the work I'm about to describe was led by Ed Burris, a former postdoc, a current postdoc in my lab. And what Ed did was compare the effect of landmass, that is island versus mainland, on speciation rate by using hidden state models implemented in RevBayes. Now, these models improve on previous approaches in 2 key ways. The hidden state model reduces the likelihood of attributing a rate shift to any given character, of falsely attributing a rate shift to any given character, and it also explicitly accounts for background rate heterogeneity across the tree. And when Ed applied this model, what he discovered was actually equivalent rates of speciation between the mainland and the islands, no special signature of faster evolution on islands. He unpacked this a little bit further and compared rates of speciation on those islands based on speciation mode, whether speciation rates differed, whether speciation was based by in situ speciation or dispersal-based speciation, and again discovered no differences. He also looked for island-specific effects. The islands differ in overall area and topographic complexity, and again discovered no major differences in speciation rate. Likewise, he recovered no exceptional rates of morphological evolution between island and mainland species. He also looked for rates of evolution differences based on speciation mode and also based on island identity, and again, recovered no differences between mainland and island anoles. Now, when compared to their putative sister lineages, Depending on your preferred phylogenetic hypothesis, this might be Corydophanids or casque-headed lizards, or bush anoles in the genus Polychrus. We still recover anoles as faster speciating. So there's definitely something interesting going on within Anolis. Now, anoles, so Corydophanids and Polychrus, like anoles, possess gular folds or dewlaps, although these are admittedly more robustly developed in anoles, but they conspicuously lack adhesive toe pads, these sticky toe pads that anoles have. And these adhesive toe pads themselves provide ecological opportunity by enhancing access to arboreal niches. Now, it's likely that these adhesive toe pads facilitated the anole diversification, although the selective pressures underlying this diversification almost certainly differed between Latin America and the islands. In Latin America, these toe pads might have facilitated diversification in the face of really strong competition and predation through cryptic evolution, or by providing ecological opportunity in their absence via enhanced resource availability or enhanced mobility on islands. In either case, these adhesive toe pads were likely a critical prerequisite for the incredible biodiversity that we observe in anoles, both in terms of their phenotypic diversity and in their species richness. But somewhat surprisingly, the islands themselves do not provide any enhanced context for this adaptive radiation to unfold. And this story, this finding that mainland anoles are really not so different from island anoles, contributes to an emerging picture, that extends certainly beyond these studies and into other labs. Now, island anoles have received a lot of attention over the past few decades, in large part because the islands themselves provide an extremely amenable setting to study their diversification. But as more evidence begins to emerge, it turns out we're finding more parallels than differences between these mainland and island radiations. Now, lastly, we were intrigued by this possibility that adhesive toe pads might function as, a putative key innovation, an idea that has certainly been suggested, and unlock more generally arboreal niche diversification and facilitate speciation. To sort of probe at this a little bit more, we capitalized on the repeated losses and gains of adhesive toe pads in geckos. And when we did, we discovered that speciation rates are uniformly higher in gecko lineages possessing adhesive toe pads than in the lineages lacking them, suggesting that this might be a more generalizable phenomenon. And with that, I just want to thank you all for being here and for giving me this opportunity to share my research with you and hopefully answer any questions that you might have. Thank you.
Thanks, Martha. That was awesome. So the format of it, I'm sure there's some people that are absolutely busting to ask some questions. But the format of this one, I think, is that we're going to switch across to Mark, and then we'll have questions at the end. So so everyone just hold still, hold on to those questions anyway, and we'll switch over to Mark now. But yeah, thanks for that, Martha. Awesome.
You can see the screen okay, and you can hear me? Can you hear me okay?
Yep.
Great.
Yep.
So thanks so much for having me. I wish I was there in person. That would be way more fun. It certainly seems that Australia is doing everything right, as well as New Zealand, and everyone else is doing things wrong. So I am there with you in spirit. Today, I'm going to be telling you about the evolution of life in the urban jungle. But because I haven't met many of you, I just thought I'd introduce you to where I'm coming from. So right now, I'm in my office at the University of Toronto, Mississauga, which is one of the 3 campuses of the University of Toronto, on the shores of the Credit River. And this is the traditional land of Mississaugas of the Credit, and before that, the Huron-Wendat. This is also the home of the Center for Urban Environments, which at the beginning, she identified that I'm the director of this center, and our vision is to discover how to promote healthier, sustainable urban environments for all life. But I haven't come to studying urban environments very easily, to be honest. For a long time, I actually didn't like cities, and I spent most of my time trying to stay out of them. But and I'm an evolutionary biologist. And so today I'm going to try and merge those 2 interests and try and answer one very general question with you. And that is, how do urban environments affect the evolution of life? You may be wondering how cities kind of relate to today's topic, the speed of evolution, and I think they are a fantastic model to understand evolution in general and rapid evolution in particular. And that's because cities can drive evolutionary change at compressed spatial and temporal scales. Now, we've known for a long time from work by urban ecologists for the last 3 or 4 decades, much of which was done in Australia, that urbanization, both in space and in time, leads to predictable changes in the environment. Both abiotic factors and biotic factors are changing as you go from these non-urban environments to these urban environments. But these same environmental factors can lead to both adaptive and non-adaptive evolutionary changes within populations. And although I've spent most of my career out of cities, I now see this as a really rich ground to ask questions of both basic and applied significance in evolutionary biology. Because when you think about it, urbanization is really just the best replicated, largest scale experimental evolution study of all time. We have literally thousands and thousands of cities, often changing the same environmental factors, and we can study whether organisms are able to adapt to that in similar ways. And of course, some cities differ in how, in the environmental factors that change, and we can understand how those differences also influence evolutionary trajectories. So as I got interested in this topic about 8 years ago now, I wanted to really understand the breadth of the topic and what had been done. It didn't seem like much had been done at the time. And so I teamed up with my good friend and collaborator, Jason Munshi South, and we wrote a review for Science 3.5 years ago. And we tried to provide a comprehensive understanding of how urbanization around the world influences evolution by identifying every single city or every single system in which people had looked at this in cities using some very strict criteria. We found that there had been many more studies than we originally thought. They've been done around the world, and in total, we found, as of 2017 in November, 192 studies on 134 species. Some continents were better represented, such as North America and Europe, but this had been studied on all inhabited continents, including Australia and New Zealand. But what was most striking is that over half of these studies were done in the last 5 years. So this is an area that has rapidly taken off. There's a lot of interest and new studies coming out pretty much every week. And since this paper has been published, it has exploded even further. So let me share with you some of the conclusions that we came away with in this review. So we found that cities can affect evolution in 4 main ways. First of all, urbanization can influence mutation rates, and this is mainly through the effects of pollution, air pollution, and water pollution and soil pollution that can influence both somatic and germline mutation rates. This is, in fact, the least known mechanism by which urbanization influences evolution. So if you want to make a big and fast contribution to this area of study, this is certainly an area that is in desperate need of more research, and it has huge applied significance as well. The second way in which urbanization influences evolution is through genetic drift, which I'm defining here as stochastic changes in allele frequencies through time within a population. It can also influence gene flow, defined as the effects of dispersal and mating on the movement of alleles between populations. And urbanization influences both genetic drift and gene flow because it causes fragmentation to natural habitats. You can see that, of course, in Australia and across all of your cities, picking here Canberra, your capital, and natural habitats such as these mountains and grasslands to the east and south and west have been fragmented as Canberra has grown up. And this fragmentation leads to predictable changes to populations. This causes population bottlenecks, especially for native species, local extirpation of some populations and species, and increased founder events. These population level consequences can then influence evolution. For species that experience these types of population consequences, they're expected to experience increased effects of genetic drift within those populations and decrease effects of or decrease gene flow between populations. There's lots of examples where we see this in response to urbanization. One of the best and one of my favorite examples, although a somewhat tragic story, is in your own backyard of Canberra. The grassland earless dragon, critically endangered, is now located only in 2 locations. The main one is in Canberra, and because of urbanization, we see much decreased genetic diversity within its populations and really strong genetic differentiation. The last way in which urbanization can influence evolution is through natural selection and resulting adaptive evolution. But this mechanism, surprisingly, perhaps, we know relatively little about how urbanization influences that. And so in the rest of this talk, I'm going to try and answer this gap. Can populations adapt to urban environments due to natural selection? We've been studying this in a number of systems. Just to give a shout out to Martha's theme of islands, we have been working on islands with this plant, Tribulus cystoides, caltrop or puncture vine. It occurs in Australia along the coastline. And we've been studying this on the Galapagos Islands, where we've been looking at coevolution between Darwin's finches and the morphology of the seeds that you can see in the top right here that are very, very spiny, if you're familiar with that, that can act as defenses. And we've been looking at how the development of towns and influx of people through tourism is altering natural selection on the morphology of these seeds. And it does appear to be actually doing that, and that paper's in review right now. But I'm not going to talk any more about that, although I do like working on islands for sure. To answer this question, I'm going to talk about one system, and that's the white clover system. And specifically, I'm going to tell you the story of the Global Urban Evolution Project, which we affectionately refer to as the GLUE project. And after this talk, if you're interested in learning more, you can go to globalurbanevolution.com. So we became interested in white clover as a model to study a number of different ecological and evolutionary questions. It's a native of Europe, but it's been introduced throughout the world. It occurs literally in every temperate Mediterranean and semi-arid city in the world because it's been transported for agricultural purposes. It grows as these low herbaceous plants. You probably all walked on it in your local towns or cities, or maybe even on your own lawn. The reason that I was initially interested in it is because I studied the evolution of plant defense. And this plant has a very interesting polymorphism, Mendelian polymorphism, for the production of an anti-herbivore defense, hydrogen cyanide. Cyanide, highly lethal to herbivores that consume it. But you find when you look in a population that not all plants are cyanogenic. Some of them produce hydrogen cyanide, and we refer to those as being cyanogenic. The other ones are acyanogenic. And this difference of whether they're cyanogenic or acyanogenic is controlled by a simple Mendelian polymorphism with 2 loci, each with 2 alleles. So that the cyanogenic plants have a dominant allele at each of those 2 loci, AC and Li, whereas these acyanogenic plants are homozygous recessive at one or the other locus for these non-functional alleles. And so by phenotyping these plants, determining whether they are cyanogenic or not, we can understand their genetic differences as well, which we've confirmed with PCR and whole genome sequencing. We were honed into this system because of classic work by Huner de Day. Huner de Day looked at this trait on a large landscape and found that as you increase in latitude and increase in altitude to colder temperatures, you see decreased frequency of these cyanogenic genotypes. And it's because not only is this a defense, but the metabolic components underlying the defense decrease tolerance to freezing. So there's selection against those functional alleles in cold habitats. We thought this same type of thing that he saw at these large scales may be happening in cities because they have urban heat island effects. And indeed, in 2016, we published the first study on this, showing that urbanization leads to repeated evolution of decreased cyanogenesis within cities. So decreased frequency of producing hydrogen cyanide. We then went on to do a number of studies and also modeling, trying to work out some of the details of this. So based on these initial studies, we thought this would be a great system, given the simple genetics, easy to find, easy to work with, and its global distribution to understand adaptation to urban environments, not just in our own cities, but on a global scale. And so what we did was we launched the Global Urban Evolution Project, where we are trying to address 4 questions. Does urbanization lead to convergent environmental change throughout the world? Does convergent environmental change cause parallel evolution in an ecologically important trait, this hydrogen cyanide? What environmental variables drive parallel evolution to cities on a global scale? And can adaptive evolution, can non-adaptive evolution, genetic drift and gene flow explain the presence absence of these declines? So to do this, of course, we couldn't go to, we could go to a few cities, but we couldn't go all over the world. So we opened this up to the community of scientists throughout the world and tried to find collaborators through listservs and social media. And we were astounded by the response. People got really excited about this, so much so that we ended up with 277 collaborators that signed on to GLUE. As far as I know, this is the largest collaborative study of evolution, including many people from Australia and New Zealand, some of which may be watching right now. And so thank you so much for collaborating with us on this. And of course, a project of this size needs a lot of leadership. And so I was very fortunate to work with all of these people that made up the GLUE lead team. All of the statistics and genomics that you'll see were done by James Santangelo, but everyone you see here has touched this project in myriad ways. And so in the end, these collaborators were able to study evolution of white clover and in this trait across 160 cities in 26 countries on 6 continents. And I think I was inspired by traveling through Australia in 2001, and everything I saw across Australia is big. You really like big things. And so that's carried with me. And so I think this is the biggest study of parallel evolution that's been attempted. To give you a sense of how we would sample, so this is Melbourne, where Gareth Hopkins and Martin Lockhart were part of the collaborating team there. They started in the city center, and every little pink purplish dot you see here was a population. And they would collect 20 plants from each one of these populations going out into the rural areas to the southwest, and they also went to the northwest to fill out the number of populations. And as people were going, they sent us pictures via social media and other means showing us they're collecting, the plants that they were seeing, the phenotyping they were doing for understanding the genotypes, the kids that were joining them for doing it, the pets that were helping them design the transects. And it was clear that people were excited about this and they were having a lot of fun. And we were all kind of working towards kind of a common set of questions using the exact same methods across all these 160 cities. And so in total, with these cities, we were able to sample a little over 6,100 populations, close to 110,000 plants, all of which we determined whether they were cyanogenic or acyanogenic. And then we had people send us tissue, and to date, we've sequenced 500 whole genomes at low coverage from 26 cities using Illumina NovaSeq. So I'm going to quickly go through some of these questions, and we don't have time to go through all of the data. So I'll give you the answers to some, and I'll show you the data to others. So does urbanization lead to convergent environmental change throughout the world? The answer to this question is very clearly yes, and I can show you the results afterwards. I'll just tell you the result here. Urban environments converge in certain features and are in fact less variable than non-urban environments, whereby you can take 2 cities in the world, Melbourne and Toronto, and they will be more similar to one another in multiple environmental features to one another than either one is to the surrounding non-urban environments around it, which is really astounding. So given that we see this convergent environmental change, we ask, does convergent environmental change cause parallel evolution in an ecologically important trait? And so what you're seeing here are the results colored for each city, where we see positive declines, negative declines, and no declines in gray. And let me give you a sense of what those look like. And I'll use Australia as the example. So if we look at Canberra, that was sampled by people from Kay Hodgins' lab, Christopher Lee, Lauren Kim, and Kay from Monash, they went from the center of Canberra down to the south and southeast. And what they found was that if you look at the frequency of hydrogen cyanide within each population represented with one of these dots, remember there's 20 plants sampled in each one of those populations, as a function of distance from the city center, which is correlated with numerous environmental factors, which I can show you later if you want, we see absolutely no change. So there's no decline in hydrogen cyanide with going to more rural areas, which are the large values here. More urban areas are at 0. By contrast, if we go to Sydney, Australia, specifically Parramatta, where it was sampled by these 3 individuals, what they see is a very strong climb where we see evolution of decreased cyanogenesis near 0 in the center of Parramatta to 80% on average in the more rural and distant suburban areas. So a stunning climb in hydrogen cyanide in that city. When we look across all 160 cities, we see that there is indeed a distribution. The vertical line here is the overall mean in this histogram, and the main effect of distance from the city center was significant. If you look at individual cities, all the ones in blue here show significantly positive climbs. So evolution of decreased hydrogen cyanide in the city center, which is exactly what we saw in Toronto. There's a few that go the other way, where hydrogen cyanide is more common in the city, and then a bunch that show no significant climbs. And each one of the cities is shown as a separate line here. So does convergent environmental change cause parallel evolution in an ecologically important trait? Yes, sometimes. If you use linear regression, it's 35% of the time. 24 hours ago, we came to the conclusion that really we should be analyzing this a little bit differently, using logistic regression, which is a better fit to the distribution of the data. And we see actually 48% of the cities show significant declines in hydrogen cyanide, but those results are only 24 hours old. So everything else is based on the initial linear models. So if we try and identify the environmental variables that drive convergent evolution in cities, I'll just tell you the result in the interest of time. We see that urban-rural changes in winter vegetation and snow are important, but it really depends on the regional summer temperature. So we get a qualitatively different effect of urban-rural changes in winter vegetation in warm cities versus cool cities. The last question I'd like to try and answer is, can non-adaptive evolution explain these climbs? So is this really adaptation, or could this be genetic drift or variation in gene flow? To try and answer this question, we have been doing whole genome sequencing of white clovers, where from each of 26 cities, we've taken 20 individuals. 10 of those individuals are from the city center. 10 of those individuals are from distant rural populations. We sequence their genomes, and then we align them and call SNPs within each population. And so we can ask, do urban populations exhibit less pairwise nucleotide diversity? And this would be consistent with greater genetic drift if in fact we saw this. And the reason that we were interested in this as a mechanism is because we showed in some modeling results that genetic drift alone, if you have greater genetic drift in cities, which often happens for many species, this can actually lead to evolution of acyanogenic frequencies increasing just by drift alone because of the epistasis among the loci that control this trait. But what we find is no difference in nucleotide diversity between urban and rural populations. And if you look at the difference in nucleotide diversity between these urban and rural populations, it's not related to the strength of clines at all. So this cannot explain our results. Do cities with clines also exhibit greater genetic differentiation, which would be consistent with the homogenizing effect of gene flow preventing adaptation? We see no difference in the strength of genetic differentiation between cities with clines versus no significant clines. And FST, the measure of differentiation that we're using, isn't related to the slope of these clines either. So can non-adaptive evolution explain the presence-absence of clines? These clines frequently evolve despite no evidence of differences in drift between urban and rural areas, and low differentiation, which implies high gene flow. So can populations adapt to urban environments? You know, the balance of evidence suggests that absolutely they can, at least close to half the time. And so the next steps is we're sequencing 2,000 genomes for more individuals to understand genome-wide patterns of selection, demography, and gene flow in response to urbanization. And we see this as a model to understand evolution in response to urbanization on a global scale. So with that, thank you so much for your time, and hopefully we have time for discussion and questions.
Thanks, Mark. That was also awesome. Utterly awesome. That is a very large study. Bravo for pulling it off. So I thought I'd open the floor for our other panelists, Luzka and Celine, to if they have any questions of you before we open the to the floor generally for the broader question.
Okay. Thanks both for fantastic talks. Can you hear me all right?
We can.
Both excellent. So just a question, I guess, for both of you. And maybe this is a little bit simplistic, but with Martha, you've got less radiation going on in the island lineages than on the mainland. And in Mark, you're saying that there's a large proportion of cities where you're not seeing change. So slightly turning the question around as rather than what's driving evolution where you see change, what's constraining it in the situations where you're not seeing it? And is it too simplistic to just think that, is it a lack of genetic variation? So with Martha, could it be that your island populations have less genetic variation than the mainland ones? And that would explain the lack of evolution rather than the behavioral inertia. And I guess I was just thinking the same thing with Mark in terms of the Sydney versus Canberra comparison. And obviously Sydney's a much, much bigger city. And so maybe the populations there have got more genetic variation in them. Maybe it's impossible to kind of both answer those at the same time, but I'd be interested in your thoughts.
Martha, you went first time, so why don't you go again?
Sure, thank you. It's a really good question. I would say that it's probably a mixture. There are certainly species on the mainland that are geographically quite diverse and also have correspondingly lots of genetic differentiation. And theoretically, those could be, that could contribute to some of the patterns we observe here. But that's not to say that there aren't, fairly widespread species as well in the islands. And those island radiations are in many cases as old, if not older, than some of the mainland radiations. And it's also the case that certain mainland species are restricted to mountaintops and, probably have lower overall genetic diversity. And so I'm not sure that it would necessarily explain, but it is a really good question that could be further explored.
Yes, so at the microevolutionary scale, at least in the trifolium system, we don't see the amount of genetic diversity, either genome-wide or at these loci, predicting whether or not we're going to see evolution of these clines. We have looked at city characteristics, so we've looked at the size of the cities, we've looked at the age of the cities, we've looked at the number of people living in those cities, we've looked at the density. None of those predict whether or not you see these clines. And the strongest predictor at this point seems to be a combination of warmer places where there are probably clines in herbivores as you go from the rural areas to the city areas. But in the north, I think it switches, it seems, and that it seems to be less about herbivores and more about these temperature gradients that these cities are creating in winter that select against the cold tolerance aspect of these traits. So it ends up being really complicated in that what drives these clines in the north, I think is very different, in our north, your south, to what drives these clines in warmer climates. And so, and when you're in the middle, I think there's just relaxed selection along these clines. So it's for this, with respect to this trait, it's just kind of drifting around along those urban rural gradients.
Cool, thank you.
Celine, do you have any questions?
Yeah, I do. I think it goes to Mark, because that's where my familiarity in terms of research context goes into. So, and maybe it applies to some island research as well. It's, to me, I've always struggled with the idea of looking as urban, or city life as having this homogeneous effect on the animals and plants that live within it. It's like saying that all islands are the same, right? And while I understand that you can say, well, that cities are more alike than their rural counterpart, what I have observed throughout my research is that you have local adaptation to really fine scale ecological conditions, and that these can vary within a city. And I just wonder whether that can maybe explain some of the strong evidence for convergent evolution, because what you're dealing with is a micro, really fine scale ecological conditions to which plants can become, adapted to.
Yeah, this is a really important question, and the question about heterogeneity in both urban ecology and evolution, and how it influences ecological and evolutionary processes is right at the cusp of our knowledge right now. I think it will surprise many people, maybe not all people, to see this result of urban environments converging so dramatically and being less being more homogeneous in their environments, so less variable than nearby non-urban areas. That was certainly surprising to me because I know, just like you, that as I go from here to my house, which is 700 meters away, there's a lot of differences, right? And surely that must also influence evolution. Well, it does. So we have looked at these smaller scale clines across. So one of the things in Toronto is there are these ravines that transverse the city. And you have, suburbs on the outside, and then you've got these really green patches on the inside. So we've just gone 2 kilometers into these. And in 3 of the 6 that we looked at, we see the same types of clines. So you do get this fine scale adaptation, or at least this fine scale evolution as well. But when you span out and look at the average effect of urbanization, you do see this overall signature as well. So I'm not saying that there isn't heterogeneity and that's not important, but it has different effects at different spatial scales at which you're looking at. Does that make sense and answer the question?
Yeah, it does. It does. I'd like to think of it as that you have archipelagos, within cities, and they're just equally as important as then you're saying, yes, you can look at one archipelago versus others. But yeah, it's.
I think that's a nice way to look at it, but I think it really depends on the organism you're looking at. So for the grassland earless dragon in Canberra, absolutely. Yeah. That thing is stuck in 3 little spots. And it's quite scary for the kind of thinking about the persistence of that species. For white clover, no, this is Pamnixia. These things are just gene flow going wild across these cities. And despite that, we still see these adaptive clines.
Do you have any anything additional to add to that, Martha, about the granularity of the sampling, or?
Well, not with relation to that question, but certainly spatial and temporal heterogeneity is something that really impacts the efficacy of thermal regulation. So I definitely found some parallels with that discussion. Cool.
I also have questions, but I'm going to put them off in favor of opening up to the floor. And we'll start with there's one from Craig Moritz in the Q&A section. He's asking whether release from predators on islands increases regulatory opportunities, and are there cases where there's introduction of predators to islands and the trend has been reversed?
Yeah, that's a great question, and that's the experiment, one of the really great experiments to run. I can think of 2 natural or semi-natural experiments that would be really good settings to put that to the test in anoles. The first is for decades, Jonathan Lassos has these experimental evolution islands in the Bahamas. Some of these islands only have an anole species, and some of these islands have a big, mean, lizard-eating terrestrial predator. For trunk ground anoles, access to low vegetation and the ground is sort of, part and parcel of their thermal regulatory strategy, and yet part of that niche has effectively been taken away from them by the presence of these ground predators, which pushes them up into the shrubs. So this would provide a good experiment to see how limitation, more so in the horizontal dimension along the sun-shade axis, might impact, by the presence of this predator, might impact thermoregulatory efficacy. A separate natural experiment occurs, is available in these dredge spoil islands off the coast of Florida. There is a much more arboreal species of anole, the green anole, Anolis carolinensis, our only native species to the United States. And in the absence of its ancestral, so this species is actually ancestrally a trunk crown anole from Cuba. It's phylogenetically nested within trunk crowns, so a relatively high perching anole. As it was released from its competitors in the United States, it actually expanded its niche downwards and becomes much more of a trunk crown / trunk trunk ground anole. Now, on these experimental islands off the coast of Florida, researchers have introduced secondarily Anolis sagrii, a Cuban trunk ground anole. And this has effectively pushed Anolis carolinensis further up into the canopy. And so it functions, I mean, it doesn't really eat Anolis carolinensis, but it's a bit of a bully, and it definitely pushes that species further up into the canopy. And so it's restricting access in this case along the vertical niche. And so that would be a good case scenario to see whether restriction along the vertical niche might, restrict thermal regulatory capacity in islands that have both species relative to islands that have only one. And I think maybe some combination of those two types of systems might be able to answer that question.
Yeah, maybe a little follow-up with Mark on that one. Is there anywhere in the world where clover is not heavily grazed by herbivores?
Well, there's certainly places where it doesn't occur, once it gets too hot and too dry. But in terms of where it's not eaten, we've looked, people have predicted that you should see less herbivory in colder climates. And this was tested by Angela Moles at University of Sydney or University of New South Wales, and found that there wasn't general support for that in general. And we looked at it in trifolium, and we also don't find support for that. So things eat this a lot of places, but the defenses are only really effective in warm climates because of the activation temperature at which, this is another, meshing with Martha, it's a kind of a thermal story. The hydrogen cyanide is only active after above 21 degrees Celsius. And so if it's below that, it's just not effective.
Cool. We might switch to a question from Ari, which I think everybody can read, but I'll read it out anyway. Ari's interested in whether acclimation, which is in turn obviously affected by behavioural responses in lizards, if they spend all their time in hot places, they have a tendency to acclimate to hot temperatures. He's wondering how much that's playing a role.
So for the data that we used, these were sort of directly measured from the wild, and we and in most studies, honestly, that have these sorts of data, there isn't really an acclimation component to them. You're usually trying to get sort of the native CT max. There is hardening capacity in these anoles, and actually there is an imbrication with Mark here, because there's been recent work on Anolis lizards by Kristen Winchell and Shane Campbell-Staton in Puerto Rico, showing that urban heat islands actually have transcriptomic signatures on heat tolerance. And so certainly there are behavioral influences, acclimation influences, and maybe even short-term evolutionary responses that may be at play.
That's fantastic work, and people should definitely look into it.
Maybe we move on to Zachary's question. He's wondering if if Martha has any thoughts on data, how different on thermoregulatory activity on the islands and on the mainland, and whether that could impact the opportunities for mating and in the long run impact sexual selection in the species.
I think that's a really good question, and I think it's definitely up for exploration. We know surprisingly little about this. I would say one of the folks who studied how movement rates relate to sexual selection in Anolis would be Ambika Kamath, and she's done that mostly focusing on Anolis sagrii. So this is, restricted to one species, but her work is extremely elegant and extremely detailed. I would say that those movement rates probably affect a lot of things besides thermoregulatory ability. So I think it's a very good hypothesis.
Cool, thanks, Martha. Is there any other questions from the floor? Got a few seconds, but it looks like we might be done with questions from the floor. The only other, well, no, maybe I'll shelve my question because we're almost out of time. Oh, okay. I'm going to ask it anyway. Temperature as a divergence, as an environmental axis for divergence. That seems to be the common thread here. And in one case, we've got behaviour kind of making it disappear in some ways, and in others, clover can behave presumably in strange little planty ways that I can't possibly observe. But is there any generality we can pull from these two talks about the role of temperature, particularly in the context of climate change and adaptation to climate change? Just that's not a question; it's a vague notion that perhaps I'm trying to elicit some response from the two of you.
I guess I can start with that because so I'm going to make the question that I think I heard, and so I think
good, perfect. That sounds good.
So, how does temperature, behavior, and behavior interact under a changing climate, for example? Okay, so I've thought about this a lot, in particular in these cybotoid anoles I've been working on for a long time in Hispaniola, because they're tropical by ancestry, they like it surprisingly warm, and their habitats are extraordinarily cold. So by all intents and purposes, I mean, it seems like, well, they should be readily buffered from environmental warming, or at the very least, maybe it's even a benefit. So one thing we did in my lab is sort of model that using biophysical models of activity. And lo and behold, these high elevation anoles that are surprisingly warm adapted for their habitat should experience, all else being equal, expanded hours of potential activity. Warming actually confers a benefit by allowing them to be out and about more often. So it would seem, at least, first principles, like this is, maybe it's a success story. Maybe this is one of those examples in which climate warming isn't always a bane. But we probed a little bit further because these high elevation anoles are sort of inexorably linked to these high montane pine forests, and they're not found outside of them. And they're parapatrically separated from their lowland competitor by the ecotone separating that high montane pine forest from lower elevation cloud forest. And it turns out that the forest fares much more poorly under warming based on our models and our understanding of the distribution of those species. So that presents actually an indirect risk of thermally driven warming and also like restrictions on water. And then we also looked and asked whether climate warming may benefit thermally the competitor species. And it turns out that it actually benefits the competitor species in relative magnitude in a greater way than the species currently occupying those habitats. So in other words, those high elevation habitats proportionately become even more thermally accessible to the lowland competitor. And so the unsatisfying story is that it's complicated and that maybe sometimes the risks may lie in the indirect effects of warming rather than the direct effects of warming.
I guess I'll take a crack at this as well. And that one of the things I appreciated that Martha said at the beginning of her talk was she's interested in why some traits evolve really quickly and others don't evolve very quickly. And I think if you think about climate change and anthropogenic disturbance, and particularly relating it to cities, one of the most predictable things that's changing, of course, with climate change and with cities is temperature. And I think if we're trying to predict which types of traits are most likely to show signatures of adaptation to climate change or anthropogenic disturbance in the form of urbanization, I think it will be these traits that are related to thermal tolerance or temperature sensitivity of some form, whether it be in plants, animals, fungi, you name it. And so in terms of, this is one of the questions we're asking right now in urban evolutionary biology is, what are the traits that are going to evolve most consistently? And I think it'll be the environmental factors that change most consistently. And the main one, or one of the main ones, is certainly these urban heat islands, and the same thing with climate change. We've done studies of testing for climate change or adaptation to climate change in white clover. We actually don't see a signature of it over the last 70 years. We do see a signature of adaptation, continued adaptation to the introduced range, but not in terms of the temperature changes that have happened since 1950 to 2020. But we do see it at least within cities. So
the answer is it's complicated, it sounds like. Yeah.
Can I just...
Celine, Liska, do you have any...
yeah, can I just jump in there? Because I think that was Mark partly answering the question I wanted to ask him as a very big, broad, general thought of, do you expect climate change to have a bigger effect on urban populations than non-urban populations? In part because the urban populations have already experienced a rise in temperature and gone through the associated selective pressures with that.
Yeah, in a lot of ways, because they're just kind of these convection ovens, right? You've got all this impervious surface that's just reflecting the sun's energy as a radiant heat. And so as you're having climate change compounded with the urban heat island effect, absolutely. And then that also causes decreased potential of evapotranspiration, increased effects of drought and desiccation and all of those things. Yep, absolutely.
Maybe putting it differently, do we have any sense of whether the kind of increased linear increase in temperature is going to have diminishing effects on a population or acceleratingly adverse effects?
I think Martha's better suited to answer that.
So sorry, I actually cut out a little bit on that question. Could you repeat the last one?
Yeah, just thinking with the ongoing increase in temperatures, which will result in even higher temperatures in the urban populations, do we expect those effects to be larger at these high temperatures or not as much in the non-urban environments?
So at least in lizards, the lizards you often find in urban environments seem to be a non-random subset of the lizards that are generally found like in an island or in a general area. And oftentimes they're kind of generalist, what we would call, like, if they're anoles, trash anoles. The anoles you seem to find everywhere. And in many cases, often relatively heat tolerant. So it depends on two factors, which is really well encapsulated by Ray Huey's 2009 paper on why tropical lizards are vulnerable to warming. The first is, how does the increase in heat, well, the first is, what's the shape of the thermal performance curve, right? Are these thermal specialists or are they thermal generalists? My understanding is that a lot of urban species are probably more on the thermal generalist side of things, although that can also vary. And then it also matters, where is the current environment in terms of how that interacts with the thermal performance curve? And how does a degree or a degree and a half matter for how much the performance detriment is? So it may be the case that and we do know that these thermal performance curves have a steeper drop off on the warm end. So it may be the case that a higher magnitude change doesn't really induce a very strong performance detriment if it's on a safer part of that thermal performance curve, or correspondingly, even a shallower temperature change can have a massive performance cost if it's really at that point where the decrement in the curve really takes off. So I guess it depends on where those urban anoles are relative to the current environment. What is the magnitude of change and how would that impact, fitness-based activities? So it's also complicated.
Thanks, Martha. I think, well, I'm well aware it's getting towards bedtime in North America. Certainly would be my bedtime. I'm an early going to bed kind of person. Mike, looks like Mark is typing an answer to you, so hopefully he can type really fast. But while he's typing, I'd like to say thank you to both Mark and Martha for coming all this way. And staying up very late to talk to us. Really awesome talks. Really enjoyed both of them. Just such cool projects. And also to Liska and Celine for joining us on the panel. It's been a great discussion. So thank you very much, everyone. Good to see you all.
For having us. This was a blast. Can't wait to visit in person again.
Yeah, thank you so much to everyone
soon.
for joining us for two great, amazing, inspiring, interesting talks. Thank you for all your time. And yeah, we'll post the recording soon.
Thank you.
Thanks, Juliana. Thanks, everyone.
Thank you.
Good night and good morning.
Bye-bye.
Bye-bye.